Hello everybody and welcome to the first Photosynthesis 2030 webinar. I'm Dr Louisa Dever and I will be your host, and I'm delighted today to welcome Dr Johannes Kromdijk and Dr Steven Driever as our first speakers. The webinar series is organised by Capitalise, an EU Horizon 2020 project that uses state-of-the-art genotyping and phenotyping techniques to exploit natural variation in key photosynthetic traits. You'll hear about some of our work and many other photosynthesis improvement projects in these webinars. Our webinars take place on the first Tuesday of each month and aim to facilitate communication and encourage collaboration across the photosynthesis community towards the common goal of improving photosynthesis in crop plants and to be able to translate this to growers within a decade. Part of the Capitalise effort is to place photosynthesis at the centre of future crop breeding research strategies and to be able to influence funding decisions. International collaborations are important routes to help translate research and to develop future-proof crops. And I'm pleased today to say that today we have a truly global audience. We'd like these webinars to be interactive. So throughout the talk, please put your questions into the question box and I will try to put them to the speakers at the end of the presentations. Presenting first is Dr Johannes Kromdijk, who is at the Department of Plant Sciences at the University of Cambridge. In the Capitalise project, he leads the work on the kinetics of photosynthetic responses and tuning the Calvin cycle. Today, Johannes will talk to us about the role of NPQ in protecting against too much light and why altering NPQ has the potential for crop improvement. I'm going to hand over to you, Johannes.
Thank you very much. Can I just check that you can hear me okay and you can see my screen?
Yes, I can. That's great.
Perfect. Okay, so we'll assume that that goes for everyone then in the audience. Thanks again for inviting me to this webinar series. It's a great pleasure to talk about some of the work that we're doing on NPQ in the context of improving photosynthetic efficiency. So my name is Johannes Kromdijk. I work at the Plant Sciences Department at the University of Cambridge. And NPQ, or the protection of plants against too much light, is one of the main areas that we focus on. But to backtrack a little bit onto the sort of greater context of this series, I just wanted to focus or kind of dwell a little bit on this population crisis that we seem to be experiencing at the moment. If you've followed the news over the last 2 years or so, you may have seen these news. This is focused on the BBC, but this has come through in many other countries too, where you may have seen that we're in a population crisis, but the bigger population crisis that would be suggested from these nationally focused crises is actually going in the opposite direction. So what you see here is that clearly a lot of countries are worried about their slowing down of population growth. For obvious reasons, that creates issues with the population distribution in several countries, and at a national level that has pretty big implications for how we organize our societies. But in the much grander scale of global population, it's worth emphasizing that what we're looking at for the foreseeable future is quite a vast increase in the number of people on our planet. So if we look at where we are today, we're approximately with, well, just under 8 billion people on this planet. And as you can see from this trajectory, which comes from the UN Population Division, which is freely available for anyone interested, they make these predictions, and they predict that by 2050, we'll be with about 9.7 billion. And you can sort of pick any preferred year in the foreseeable future, at least, and you can see that the trend is still going up, with the greatest uncertainty here being our own productivity, which is indicated by these blue dotted lines, which is plus or minus half a child. So I guess in the context of this seminar series, what we're really trying to look for is how can we provide enough food for all these additional people in years and decades to come. That's somewhat familiar territory for at least sort of relatively recent history, where Thomas Malthus, who's a famous politician in his own days and a famous Cambridge academic, he's mostly remembered for stating that population will always grow more rapidly than food supplies until the numbers are reduced by war, disease, famine, or any other type of disaster one can think of, I suppose. The reason that he's mostly remembered for this statement is the fact that we like to refer to this statement when we want to emphasize the power of our technology, which largely could be our reason to allow us to outpace this prediction in many ways. That's shown in this graph here by Ray et al., where over the last 5 decades, and I should say that the last decade from present time is missing because this paper is getting a bit aging already, but you can tell that the marginal gains of some of our major commodity crops have clearly been on the rise over these 5 decades. And that was great news, of course, because it allows us to sustain this growing world population. Now, if we're looking into the future, it becomes a bit more treacherous territory, I should say. So what this paper did was to look at, first of all, what levels of productivity would we be ending up if this increasing marginal gains trend would be extrapolated into the future based on the last 2 decades in the past. And so that's what you see with the solid lines in this figure. However, what they also looked at was if we look at the predicted increase in food demand by 2050, where should the productivity levels be going? And that's what you see by the dotted lines. And even though there's quite a big uncertainty range around these lines, which is indicated by the sort of faded colors behind the lines, you can still see that in all cases, the dotted lines are quite a bit higher than the solid lines. So the projected gains, even if we manage to keep the rate of improvement going based on the last 2 decades, which would already be quite something, it's still going to fall short by quite a margin compared to what is calculated to be the increase in food demand in only a few decades from now. So that means that we have to think out of the box for additional strategies to increase the productivity of our crops. And photosynthesis is definitely one of those strategies that we think is still underutilized. So what you see here is an analysis. It's essentially an accounting analysis based on the amount of solar energy coming into our planetary system. And what this analysis is doing, it's accounting for, it's an energy funnel all the way from solar energy into plant biomass. And what it's accounting for is all the losses that are deemed to be absolutely unavoidable. And so that's what you see here on the red, in the red text. So there's certain parts of the spectrum that can simply not be utilized by photosynthesis. Certain parts of reflection and transmission that is unavoidable, and so on and so forth. There's a little bit of a difference between the different photosynthetic pathways. So C3 plants have a slightly lower efficiency in this calculation than C4 plants, which utilize a carbon concentrating mechanism to increase the amount of CO2 available for photosynthesis inside their leaves. But in general, you see that the efficiency based on this calculation would be around about 5 to 6%. Now, the interesting thing here is that crop canopies, even the most productive canopies we can find, typically achieve only about 30% of this theoretical maximum, which suggests that there might be some room for improvement unless something else is incredibly limiting to this efficiency in a way that's not accounted for here. And so one might wonder, why is that discrepancy so large? Well, one big reason for it is that photosynthetic efficiency is not always at its max capacity. It's actually downregulated when leaves receive a lot of light. So what you see here is a schematic which depicts the rate of photosynthesis as a function of the incident light intensity on the x-axis. So with more light, photosynthesis initially increases and then saturates towards its max capacity. However, leaves keep absorbing light at a fairly linear rate. Of course, if they change leaf angle, that might change, but in this very simple schematic, you can see that the absorbed light is essentially increasing linearly with the amount of light incident on the leaf. And so that means that once the energy dissipation through photosynthesis is starting to saturate, a lot of excess energy is being accumulated in this leaf by simply absorbing too much more than can be processed through photosynthesis. And so if you extrapolate this into the highlight area, you can see that this really amounts to quite a substantial amount of energy that's being absorbed but cannot be used. And just like probably any one of us listening to this webinar is probably used to, it can be quite damaging to stay in too much light for too long. Fortunately, we're not limited to where we are. We can move out of the sun and find a more comfortable place, but that doesn't really work for plants so well. So they have to figure out different ways to dissipate this energy in a safe way. And so that's shown in this very dense schematic that I'll tease apart a little bit. So what you might remember from photosynthesis lessons in high school biology is that it's essentially utilizing the light energy to split water. So the oxygen liberated comes from water that is split. At the same time, that action also results in liberation of protons in a confined space in the chloroplast. And that leads to an acidification of that space in the chloroplast, which is called the lumen. So this acidification of the thylakoid lumen is a very early signal for light that's being used in photosynthesis. Those protons would normally dissipate through the action of ATP formation, through the ATPase. But if that doesn't keep up anymore because photosynthesis starts to saturate, we see an acidification of the thylakoid lumen, which then leads to all sorts of action to try and reduce the efficiency of energy used through photosynthesis by thermal dissipation. One of the things that happens is the protonation of a subunit of the photosynthetic antenna complex that you see here, the photosystem II antenna, which upon protonation starts to induce or facilitate a form of MPQ photoprotection, which allows a very rapid induction and a very rapid relaxation of that process. So a fast response type of photoprotection. On the left-hand side, you see a pigment cycle. So we've got two types of pigments. There's actually one in between, which is a sort of half in between product of these two, with a reversible cycle in between. So again, upon acidification, we see that this reversible xanthophyll cycle is starting to run in the direction of zeaxanthin through the action of violexanthin deoxidase, which becomes a lot more active under acidic conditions. Whereas under neutral conditions, the cycle reverts back to violexanthin via the intermediate enteroxanthin. And so the accumulation of zeaxanthin, or these deepoxydated xanthophylls, also corresponds quite strongly to the upregulation of NPQ, or photoprotection, in the PS2 antenna, which allows the energy that accumulates in the antenna to be dissipated through essentially thermal release in a harmless way. So it's a controlled dissipation of excess energy. Now that schematic was making things quite easy and easy to look at, but of course a crop canopy is anything but simple and doesn't conform to those very simple schematics so much. So what you're looking at here is a look through a soybean canopy from below, and it's quite interesting to look at the different types of leaves that you see in here. There's leaves that have clearly a very low light level to operate under and might barely break even for their photosynthesis. There are some leaves that might just get enough light to operate photosynthesis at its asymptotic plateau, but not much more. And there's clearly some leaves at the top which are completely oversaturated with light and would need a lot of MPQ to safely dissipate the excess energy that they receive. And so that's sort of the rationale for looking at MPQ as a means to improve photosynthetic efficiency, because we have this diversity, and this situation is constantly changing over time as well. And if we think about what might happen over time, is that the MPQ levels are constantly adjusting based on the light intensity that the leaves receive, but they can never be as quick as the light levels themselves. So what you see here on the left-hand side is a depiction of the situation where MPQ is constantly lagging behind changes in light intensity. So the yellow bars here are a schematic of a light intensity change, and the red bar is MPQ trying to keep up with those changes. You see, because of this lag time, there are areas where there's too much light relative to the level of protection via MPQ, which happens both at the start and the finish of the schematic. But there's also areas where there's too much dissipation of energy, where it could actually be utilized through photosynthesis. You see where these areas where the red line is clearly higher than the yellow bars. And so what could be more efficient is the situation on the right-hand side, where MPQ is more quickly to respond and react to changes in light intensity, and therefore more closely follow the light intensity profile with the benefit of having less time spent underprotected as well as less time spent overprotected. And so model calculations have been, quite elaborate model calculations have been used to estimate how much this more efficient situation might help in terms of carbon fixation by a crop canopy. And it was quite a large number. As you can tell, it's dependent somewhat on how chilling tolerant or chilling susceptible the species was that the model calculations were parameterized for. But especially under somewhat cooler conditions, but even under the warmer conditions, we see there's between a 10 to 30% reduction in carbon gain due to the slow responses of MPQ. And so in my research, we're trying to look and see if we can speed up MPQ responses to claw back some of this difference. And so if we look at that schematic again, what we tried to do a few years ago, what turned out to be quite successful, is to overexpress this PSPS subunit to increase this faster form of MPQ in terms of amplitude, as well as increase both of the enzymes in this reversible pigment cycle to speed up the changeovers in this cycle and also return the cycle back to the epoxydated state more rapidly when neutral conditions are returned. So in essence, what we're trying to do is make MPQ faster with these changes. And so measuring MPQ as a function of light intensity, we try to do this both under constant light intensity, which you see here called steady state. And so what you see here are three plant lines that were modified to have faster MPQ, and then the gray symbols are the control line. You see they're relatively inconsequential in how they're modified under steady state conditions. So they're very similar to the control line on the constant light conditions. Whereas on the fluctuating light conditions, we see that they're still reaching a level of MPQ under high light, which light intensity is here on the x-axis again. But so under high light, the level of MPQ is very similar, but under low light, we see a difference opening up where the control line is sustaining a lot more MPQ than the modified lines, but only under the fluctuating light conditions. And so this, we then predicted, would probably lead to a change in photosynthetic efficiency, which is what the modeling predicted. And indeed, when we measured photosynthetic efficiency, we could see that on the fluctuating light, the efficiency of the photosynthetic light reactions, as well as the light independent reactions downstream, so the Kelvin cycle in particular, we could see that CO2 fixation efficiency was improved in these modified lines as well as the light reaction side. So they appear to be more efficient under fluctuating light. And so, of course, we were then interested to see whether this would also affect their productivity performance. So these plants were grown out in the field. This was done in Illinois, the University of Illinois in the United States. And we grew these lines side by side in a replicated block design. And indeed, the modified MPQ lines showed an increase in pretty much any size measures that we could find, as well as the dry weight content, which is also the real gold standard measurement. And it also appeared that the leaf or the weight increase was distributed equally across the different organs. So that was actually quite a great surprise to us, because usually model predictions, especially if they're a bit outrageous, you think the model simply missed something. But in this case, it seemed to conform exactly to what the prediction suggested. So to summarize this, we saw that MPQ transiently does limit CO2 fixation on the fluctuating light. And the combined overexpression of these three genes gave us faster MPQ, more efficient photosynthesis, and an increase in total dry weight, which ranged between 11 to 20%. It is worth mentioning, though, that this seems to be quite specific to the species that we used. So we used tobacco, as you could see from those pictures earlier, which is a useful model species to grow out in a field and see how it works under relevant conditions. This has now been repeated in Arabidopsis more recently, and that actually seemed to negatively impact Arabidopsis, which is a small model species with a rosette shape. So it's possible that the canopy architecture that corresponds to different species may have a big impact on how well they respond to this treatment or this specific modification. We did look at one of the genes in specific, PSBS, this subunit of the PS2 antenna, which was suggested before to maybe carry most of the benefit on its own. But in this case, we actually didn't see any beneficial effects of either less or more of this gene or of the product of this gene. As you can tell here from the white bars, which have a lot less PSBS, and the red bars are lines that have a lot more. And on the very similar field conditions, none of these manipulations actually gave us a benefit in productivity. But again, this could also be species specific, because around about the same time, a group at Nottingham was doing the same type of experiment with rice, and they actually found that for rice, it increased canopy CO2 fixation to have more PSBS because of the additional photoprotective capacity. What we did find was totally unexpected, and that is shown in this figure, which I'll talk you through quickly. So what you see here is the CO2 fixation rate relative to the control, stomatal conductance relative to control, which is much higher in this knockout line compared to the three lines which have more PSBS. And as a result, this divided by this gives us water use efficiency of CO2 fixation, which shows a trend with this PSBS abundance. That was totally unexpected, but we then dived into the literature, and we noticed that this well-known response of stomata to light might be affected by PSBS. So stomata are microscopic pores on the leaf surface which respond to light. So when we shine a bit of light on the leaf, stomata tend to open and then allow gas exchange like CO2 to get in, oxygen to come out, and water vapor comes out as well. And of course, when we remove the light, then they tend to close again. The interesting thing that we found was this opinion paper by Florian Busch, who suggested that this opening response might at least in part be controlled by a component, the redox state of an electron transfer component in the chloroplast called plastoquinone. And so this schematic here is essentially summarizing what he proposed in this paper. Interestingly, this schematic would suggest that there is a role for PSBS in controlling stomatal opening, namely that the excitation pressure, so the energy going into photosystem 2, affects the plastoquinone redox state quite strongly because it's immediately downstream from PS2. And of course, MPQ limits that excitation pressure, which fits in exactly with what we saw in the sense that PSPS overexpression lines keep this pool more oxidized and therefore more closed, stomata, and less PSPS containing lines would make this pool more reduced and therefore stomata would open more. But this is still very much work in progress because we have no idea how this signal transduction might work. We did notice that if we insert this mechanism into models for stomatal conductance, we do get a better predictive ability. So again, circumstantial evidence that this might hold some value in exploring further. And that means that using MPQ modification surprisingly may also change the situation that you see here to some extent. For those of you that are perhaps not familiar with what you're looking at, this is a top-down view from NASA Earth Observatory website of center pivot irrigation in the Rocky Mountains rain shadow. And you see that this productivity in these areas is remarkably limited by the amount of water that's available, which often leads to very unsustainable situations. So if we can make crops more water use efficient through the manipulation of MPQ, that would definitely be very beneficial too. So, as I said, we don't think all three genes are necessary in all crops, but in our case, we could definitely not repeat the results with PSPS alone. Our continuing work is now also really branching out into finding out whether we can find genetic variation in MPQ traits without necessarily coming in to modify them. So in existing populations that could be used for breeding purposes. And this is where the Capitalize program is coming in, where we've got populations for some of the major crops, barley, maize, and tomato, where we're trying to figure out if natural variation has already resulted in genetic loci that could be selected for in breeding programs and achieve similar type of benefits that I've just explained in our proof of concept work using genetic modification. So to conclude, photoprotection is vital for plants to deal with excessive absorbed energy, which can easily get up to 4 times their photochemical capacity, and so it needs to be harmlessly dissipated. Altering photoprotection traits has significant effects on crop performance, and potential for crop productivity gains can come from kinetic adjustments, so making it more speedy. Could also come from increasing the photoprotective amplitude, as we've seen from some of the results in rice from another group. And we also found some surprising feed-forward effects on water use efficiency. And currently we're exploring the genetic variation for these traits across various populations for major crops to see if we can perhaps select for improved MPQ traits in future breeding programs. So with that, I'd just like to mention, thank my group, which you see here in the picture, and of course the funding from the Horizon 2020 funding, and the other funding source that needs to be mentioned here is the Bill Melinda Gates Foundation, the Foundation for Food and Agricultural Research, and UK Aid, which supported some of the work that I showed at the start through a subgrant through the University of Illinois. So with that, I'd like to conclude my webinar.
Thank you very much for a very interesting talk. Do we have any questions? Would anyone like to put some questions in the box? In the meantime, I have some questions. Is there a cost to altering NPQ relaxation? Or do you foresee any cost? For example, in less stressful conditions, do you think you might see a cost?
Well, that's a very good question that we don't really have a good answer to just yet. I think and there is definitely a possibility that it has an interaction with plant defense, and so the lipid peroxidation reactions that happen when plants get essentially over-reduced electron transport chain have, I think, evolutionarily obtained a signaling function as well. And part of that signaling function seems to instigate a response to herbivory, for instance. And so it's possible, and there's some sort of circumstantial evidence from previous work that manipulating MPQ may interact with that signaling to respond adequately to pests and predators, in which case that would need to be adjusted as well to keep things in check again. It's really quite tricky to assess at first instance. In terms of a more straightforward penalty to think about MPQ, I think if you have more MPQ than is required under steady state conditions, so if you simply increase the amplitude, it's foreseeable that under especially light limited conditions, so deeper inside the canopy, there should be a somewhat penalty on carbon gain as a result, if you simply increase the amplitude. But then it becomes a trade-off between how easily will these leaves get photo damaged under the conditions these plants are grown under, and if you manage to avoid that with a much more reversible inhibition through MPQ, it may still be beneficial. And so that's much more of a situation where it depends on the species and the type of canopy architecture that comes with that, as well as the production environment that those plants are being growing under.
Okay,
so it's a bit of a hand-wavy answer because it's not very easy to give a much more concise answer. A lot will be subject to more testing.
Is there any evidence whether it's an adaptable trait as opposed to what with variation?
There is definitely a lot of adaptation potential in MPQ. So the PSBS protein does respond to more stressful conditions, so it typically gets upregulated. The xanthophyll cycle enzymes are also regulated to some extent, as well as the pigment pool itself. Well, I think in agriculture, what we're looking at is a more protected system than in natural vegetation, where the plants in general evolution, you could say, it tends to err on the side of caution. And so I think what we're looking at is essentially shifting the balance slightly away towards perhaps a slightly more risky situation for plants to become more productive at the expense of being overly cautious. And I think you can get away with that in a slightly more protected setting like agriculture can be. It isn't always true, of course. We know of agricultural situations where it's definitely quite stressful to the plants as well. But in some cases, I think there's scope to shift that balance a little bit.
Okay, thanks very much. And with that, I think we will thank you, Guane, and we're going to move on to our second speaker, which is Dr. Steven Driever, who is an assistant professor at Wageningen University. His expertise in crop physiology, photosynthesis, stress tolerance, and crop growth models. Today, he's going to talk to us about how to estimate and exploit improvement and natural variation of photosynthesis traits for improving crop yield. Okay.
All right, thank you very much, Louisa. Just checking that you can all see my screen.
Yeah, looks good.
Okay, great. Yeah, well, thanks for joining this webinar, and thanks for the organizers for inviting me. Today, I'd like to talk to you a little bit about photosynthesis in a slightly larger context, which Juan already alluded to, which is in what photosynthesis does in a crop. Yeah, so this already is shown here in the slide. We're going to slowly move up. Let me just give you a laser pointer. Move from the leaf level up to the whole crop and see what that entails. Before I start this presentation, I'd like to acknowledge a few people, because not all of the things I'm going to show you are my work. They are done in collaboration with the people here in Wageningen: Sinjin, Paul Struik, Jochem Evers, Alejandro Morales, Peter van der Putten, and Mogus Retta, who are all part of our group, and previous PhD students Herman Berghuis, Chuan Kai, and current PhD student Bei Dong. Together also with people from Leuven, Graham Farquhar, and Javier and Ula from University of Western Sydney. Well, Van already alluded to the fact that we have a big challenge, and we have a challenge to feed a growing population. And not only do we need to feed a growing population in terms of the number of people, but also in a changing environment. So we know that temperature and CO2 are likely to rise, and that weather extremes are more likely. Now, luckily, there have been some recent improvements or transgenic manipulations that have resulted in improvements in photosynthesis. And that's really promising, because that shows that we can make crops utilize their light much better. And of course, as Juan already mentioned, there is existing natural genetic variation in photosynthesis that allows us to explore and exploit those features more. However, getting from a proof of concept or a certain crop variation, a certain crop to a more sustainable and improved yield, there are quite a few steps in between. And up to now, we've seen really nice evidence that through physiological analysis, these increases in or improvements in photosynthesis can be realized, and that's really promising. However, the level of complexity that we have at a crop scale is a little bit beyond the comprehension of what we can physiologically analyze. So there we need a tool to integrate what happens in the environment, what kind of variation or manipulation can occur, and how that physiologically interacts. So that's where that's where a physiological crop model can come in to integrate those variables and see how they respond. So even so, like a question to what is the cost of a certain manipulation, and does it hold, or is it beneficial under a number of conditions or in various crops, is something that is a question that can be asked using a model like this. Moreover, once we capture such variation in a model, parts of the model and even multiple parts of the model can be used to find genetic markers. Ultimately, this hopefully will result in an improved yield and a more robustly improved yield under conditions that we expect there to be in the future. Now, the way that we approach this is by understanding this from a bottom-up approach. And therefore, we need to scale photosynthesis all the way from the cell up to the crop. On the one hand, we need to understand how photosynthesis works. And on the other hand, we need to understand how a crop works. So basically, we need to move up from the cell via the plant up to the crop. And of course, we can all model that. But that's not where, and as well, I think Vanne said that very nicely. Models tend to sometimes miss something, so that's where experimentation comes in. And I think that I will show you a few examples of how the combination of experimentation and modeling can help us bridge those integration levels. So let's take the first step first. And let's move from the cell to a whole leaf. And to begin with the best known model, the Farquhar van Cameron-Barry model is a very nice biochemical model, which actually doesn't only work at the cell level, but also at larger scales. And this model has been around for a few years and has also been extended, because as this is a biochemical model, it does very well in describing the CO2 assimilation. But obviously, light utilization depends on the light reactions, linear, cyclic, and pseudocyclic electron transport. And here we see a nice schematic of how that electron transport occurs. And then we have photosystem 2 here that splits water and basically forms the first step into the electron transport chain via a number of electron carriers, goes to photosystem 1, where it's excited again by light. And at the end, NADPH and ATP are formed, which form the chemical reducing equivalents that are utilized in CO2 assimilation. Now, apart from CO2 assimilation, there is also photorespiration and a number of other reactions that directly or slightly more indirectly utilize this stream of electrons. So in a way, this addition to the simple Farkman-Cameron barium, or I say simple, it's not so simple, but to this biochemical model, now gives us the tools to ask the question, what happens inside the leaf? And this is a very nice example of how you can model the light and the dark reactions together, and ask yourself what happens inside the chloroplast, inside all the green parts that you see here in the picture. If you want to have a nice overview of how this model works and how it's evolved to the state it is now, there's recently been a nice review out that's worth a read, I think. Well, if we look more closely into the leaf, a chloroplast doesn't only live on its own, it lives inside a structure and with many more chloroplasts. So if we look, if we apply a biochemical or electron transport model to each chloroplast, we can reconstruct the inside of, in this case, a tomato leaf, where the green parts are the chloroplasts, where the yellow is the cytosol and the blue is the vacuole, and here's the upper epidermis and the lower epidermis. We can then look at what happens in terms of light absorbance and how much light is reaches each chloroplast. So as I was explaining about difference in light absorbance, and that the chloroplasts in the upper palisade cells receive a lot more light than the ones in the lower side of the leaf, which you can see very nicely here in this graph, where with depth the fraction of absorbed light is much higher in the palisade cells than in the spongy mesophyll cells. And that has consequences for how much CO2 is present throughout the leaf. So here we see two images of CO2 distribution. So blue is a low CO2 concentration and red is a high CO2 concentration. And as you can see, if there is photorespiration going on here at 21% oxygen on the left, then the distribution of CO2 is relatively homogeneous. And this has to do with the fact that photorespiration releases a little bit of CO2 as well. However, if photorespiration is removed at 2% oxygen, you can see that there is far less CO2 available in the palisade cells than in the mesophyll cells. So in a way, the chloroplast operating here at the upper side of the leaf has quite different environment to the ones that are operating in the lower side of the leaf. And if we then look at different structures of the leaf, it also means that the photostatic capacity of the leaves in the palisade cells, so in the upper side here, have a higher photostatic capacity than the ones in the spongy mesophyll. And that means that chloroplasts in the top of the cells really have are quite different from the ones in the bottom. As you can see here, this can be quite dramatic. So while the fraction of photostatic capacity remains really high in some palisade cells, it may be very low or even close to zero further down in these cells. Now, this is something that of course could be optimized. And if we look at what is the optimal distribution of photostatic capacity through the depth of a leaf, and here it's been calculated for 3 scenarios. So one scenario is where there is a uniform distribution, so the palisade and the mesophyll cells have an equal photostatic capacity, or an optimal distribution where there is a red line here, where there is relatively a gradual decrease of photostatic capacity, or a distinct difference between the photostatic capacity of the palisade and spongy mesophyll cells, which is the blue line here. A rapid drop when we move from the palisade into the spongy mesophyll cells. And indeed, if we look at the response of assimilation rate to CI, to the internal CO2 concentration, we see that the scenario for the optimal scenario actually has a higher maximum photostatic capacity than the other two scenarios. And indeed, the measured data suggests that scenario 3 is actually closest to the one that was observed. So there's room for improvement there. And just to show you, a similar exercise can be done for C4 plants, because obviously the last results were shown for tomato. But here as well, a 3D reconstruction can be made with a light distribution to show where the majority of the photosynthesis happens in the bundle sheath cells, and how much of that is influenced by the way that the light is distributed through the cells. Now, and that is actually shown here as well with a high CO2 concentration here in the bundle sheath cells. And this is very nice, I think, of the intercellular airspace. There's also a difference observed in the CO2 concentration, but that's as a side note. Now, let me take you from the leaf up to the plant level. And there's some striking similarities here with what I just showed you at the leaf level. This is a very nice study by Tori Clark, where she showed that with leaf position, so 1 being the youngest leaf and 10 being the oldest leaf, the structure of the leaf changes. This is obviously not surprising, because leaves grow and expand, but it certainly has consequences. And it has consequences for its photosynthetic capacity. And it seems that this photostatic capacity, if we move from young to old, decreases with leaf nitrogen, and not surprisingly also with the number of rubiscocytes. As you can see here, they very much are directly related. So leaves or plants tend to change their photostatic capacity with age, and also with the way that they invest their leaf nitrogen. So if we scale from a leaf to a canopy, or to a plant, in this case here, 3 things are important. One is the canopy size, the leaf area index, how much leaf area there is. The canopy configuration, whether leaves are perpendicular to the incoming light, or that they are more upright, so that more light can penetrate to the lower leaves. And whether the canopy nitrogen profile, the nitrogen called the nitrogen extinction coefficient, is related to that. If we then look at what the gross canopy photosynthesis does versus irradiance, is that, and here it has been simulated, that a non-acclimated canopy, so where the nitrogen profile is not acclimated to the light profile, actually has a suboptimal gross photostatic canopy rate. And if they are acclimated, then that results in a higher gross photostatic canopy. And in fact, this is true not only for C3 plants, where we see here that the fraction of acclimation decreases with day length. This is also true for C4 plants. So this seems to be just like the photostatic capacity within the leaf seems to acclimate to the light level, needs to acclimate to the light level. We see that happening for the light and the nitrogen level within height of a plant. Now, to show you that this doesn't only hold for light, there's been a study here by a former PhD student of Xin Yu Yin, that the acclimation through leaf content is influenced by CO2 concentration and temperature. So here you can see in a rice crop grown on a face site or T-face site where the CO2 and the temperature are manipulated, and the distribution of canopy nitrogen content among the individual leaves largely determined the acclimation of the canopy photosynthesis to these environmental conditions. As you can see here, there are a number of photostatic parameters. Here on the top left, there's the respiration rate, the maximum rate of RUBP regeneration, Jmax, the maximum carboxylation efficiency, which is VCmax, as well as mesophyll conductance and a light use efficiency parameter called kappa2LL, which all are related to the leaf nitrogen content, and that's where the different treatments, so whether there's been additional CO2 given, so C+, or additional temperature, or both, they all follow this this relationship with the actual leaf nitrogen content. Now, knowing that the plant can acclimate to its environment in terms of photosynthesis, let me now take you from the plant up to the canopy. And how dense a canopy is. And this is a simulation, a 3D simulation of a wheat canopy. Really depends on how close the plants are together and how much light is absorbed by this canopy, how much light is penetrated to the lower leaves. But let's not forget that the canopy itself is not the only part that the crop has to deal with. There is also competition for resources, and especially also below ground. So intra-canopy resource competition is certainly something that can also influence the above ground parts, and with that, the availability of nitrogen. Because if you see here a very nice 3D simulation of an individual bean plant, the roots have all the freedom to move around in the soil and have all the resources that they encounter for themselves. And the same for a maize plant here. However, if they are grown in a closed canopy, the roots interact. They compete for what is in the soil, and even if there are two crops grown next to each other, they tend to compete between each other. So what is a functional trait then for the photostatic? What is a photostatic functional trait then? And that very much depends on the type of crop or the type of crops that are grown in what crop system, also in what growth phase they are, whether they are just forming leaves and trying to increase their leaf area index, or whether they are seed fitting. And what the environment is at the time of that growth phase. So, I think yeah, here we are. To give you an example of how we look at those, to try to disentangle those photostatic traits, here is a study by one of Jochem Ever's PhD students, where we look at photostatic traits of maize, whether they're grown in just maize, so sole maize, whether here in orange they are grown in combination with bean, and in green whether they're grown in combination with wheat. So here's a 3D simulation of that. Obviously these measurements were done on real plants, but to give you an idea, so this is an intercrop with maize and bean, and here we see an intercrop with maize and wheat. That's how they look. So there are plants that are living in the middle of the row, and there are plants that are at the border. And when we compare the plants grown at the border versus the plants in the row, then we see that there at saturating light intensity, there are actually differences in leaf photosynthesis at a number of developmental stages, which you see here in the bottom. Here is the specific leaf area, and here's the specific leaf nitrogen content of these leaves. The gray bars represent maize grown on its own, so only maize. The orange is maize together with bean, and the green is maize together with wheat. And here for 5 different development stages, these parameters have been measured. And as you can see, that some of the photostatic parameters change obviously with developmental stage, but also whether these plants are grown either as a border or in the middle of a row, and that changes with developmental stage. Now, as you can see, specific leaf area also changes with developmental stage, as well as there's a difference between border and middle of the canopy. And in terms of leaf nitrogen, you can see that when specific leaf nitrogen, for instance, here decreases, the specific leaf nitrogen here increases, and that levels out that maximum photosynthesis rate, as you would expect. So this is still ongoing work, and this is done by Dong, a PhD student from Jochem Evers, and we're just trying to scratch the surface here of how plants behave and how their photosynthesis behaves in intercrop systems. So I hope that I've shown you here that going from the cell level up to the canopy and the crop level, that it is more than just the sum of the parts. And the way that we approach this is really to try to capture that complexity into genotype by environment crop models, sometimes additionally genotype by environment by management crop models, to identify those functional crop traits that are relevant for photosynthesis. Now, we really try to do this in a way that we use experimentation to parameterize the model, but then also to use this as in its entirety to find multiple genetic markers so that we can exploit and improve exploit the improvements that have been made already transgenically, but also tap into the natural genetic variation, which might otherwise be underutilized. So to realize improved crop photosynthesis, we think it's important to identify multiple markers for complex trait at the same time. We think our combined experimentation modeling approach is really key to that, because it allows you to then with the mechanistics of such model and the understanding and insight that it gains, allows you to design a crop ideotype for a number of trait combinations that either can maximize photostatic improvement, or to design or give direction to the kind of traits that are required for future environments and types of measurement. So with that, I hope I've given you an insight of what we do at the Center of Crop Systems Analysis. And if you have any further questions or want to know more, then these are the website, and we are also on Twitter. And with that, I'd like to thank you for your attention.
So I'm going to thank Stephen very much for his presentation. So I'd like to thank the speakers for some very interesting talks. Unfortunately, I'm not going to go to questions for Stephen. That's all we've really got time for. Next month's webinar will be featuring Professor Elizabeth Carmer-Silva from Lancaster University and Professor John Cushman from the University of Nevada. Check out the website for more information. Registration will be opening shortly, and please note the time is different. It's at 1400 hours CET. We'd love to hear any comments or suggestions you have for the webinar series, so please send us some in the feedback if you can. I look forward to seeing you all next month. Goodbye.